The physical foundations of biochemistry are thermodynamics and kinetics. We will start with thermodynamics, which is the study of whether a reaction or a process is favorable and occurs in the direction written spontaneously. All processes, including biological processes, are bound by the laws of thermodynamics. We will focus on the first two laws. The first law states that the total energy of the universe remains constant. The second law states that the total entropy, or disorder, of the universe is continually increasing. The expression that unifies these laws looks like this. Hopefully this equation looks familiar. Delta G is the change in the Gibbs free energy. Gibbs free energy is a measure of the total energy of the system, and you can think about it as a measure of the energy or work available in a particular reaction. The sign of delta G predicts the direction of the reaction in the spontaneous direction. For example, if delta G is less than 0, is a negative number, the reaction is exergonic and will proceed in the direction it is written, in the forward direction. If delta G is positive, the reaction is said to be endergonic, and the reaction will proceed in the reverse direction. Delta H is the change in enthalpy, or heat. Another way to think about the change in enthalpy is the difference in the bond energies between the reactants and products. If delta H is negative, the reaction is said to be exothermic, meaning that heat is released. In other words, the bonds that are formed in the reaction, because bonds forming gives off energy, the bonds that are formed are more stable than the bonds that have been broken. If delta H is positive, the reaction is said to be endothermic, meaning that heat is absorbed. Another way to think about this is that the bonds that have been broken are more stable than the bonds that were formed. This would require an input in the net energy. When delta H is negative, this helps delta G for the process be negative. Remember that when delta G is less than 0, the reaction is exergonic and occurs spontaneously in the direction written. Therefore, a negative delta H helps a process be favorable. The last term is T, temperature, times delta S, the change in entropy, or disorder. When delta S is greater than 0, a positive number, it means the entropy is increasing. When delta S is less than 0, or a negative number, it means there is a decrease in entropy. Because of the negative sign, a positive delta S helps to make delta G more negative, making the process favorable. This effect becomes larger as the temperature increases, because the T delta S term gets greater. What does all this thermodynamics mean for biological systems? Well, because of the second law, that the total entropy of the universe is constantly increasing, this means that systems, including biological systems, tend towards disorder. Living organisms need order. They need complex biomolecules to function. Therefore, they require an energy input to synthesize and maintain their complex biomolecules. If the maintenance and synthesis of these biomolecules requires the input of energy, in other words, these are endergonic processes. In other words, they're unfavorable processes. Cells, therefore, use energy coupling to couple these unfavorable processes to favorable processes in order to cause them to occur. In other words, cells couple an unfavorable process, the maintenance or synthesis of complex biomolecules, to a favorable process, such as hydrolysis of ATP. And because it couples these two reactions, it causes the work of synthesis or maintenance of complex biomolecules to be done. The way that cells do this is because delta G, free energy, is additive. Now let's look at a chemical example. If we couple an unfavorable reaction, such as reaction 1, the synthesis of a biomolecule, specifically glucose 6-phosphate, to a favorable reaction, such as reaction 2, which is the hydrolysis of ATP, when we couple these two reactions, their delta Gs are additive. Reaction 1 is endergonic. Reaction 2 is very exergonic. It is so exergonic that energy output overcomes the energy input required for reaction 1. Therefore, the coupled reaction, the synthesis of glucose 6-phosphate along with the hydrolysis of ATP, has an overall exergonic free energy release, making the overall process favorable. As mentioned earlier, delta G is a measure of the spontaneity or the driving force of a reaction. It can also be thought of as a measure of the amount of work that can be done by a reaction. Therefore, it's useful to be able to calculate delta G. We can calculate delta G using the following equation. Delta G can be calculated using delta G naught, or delta G 0, the gas constant, the temperature, and the reaction quotient. Delta G naught is an arbitrary reference point. It's called the standard free energy change. It's called the standard free energy change because it's always under the same set of conditions. All concentrations are 1 molar, all gases are at 1 atmosphere of pressure, and the temperature is 25 degrees Celsius. Obviously, most of the reactions we look at do not have those conditions. However, they have been chosen because delta G can be measured and tabulated under these conditions. We can then use these tabulated numbers in order to calculate delta G, which is the driving force of a reaction under any conditions. Delta G tells us the driving force of the reaction under any set of conditions, and therefore takes into account the standard free energy change plus any differences due to a difference in temperature or a difference in concentration or pressure. It does this through the other terms in the equation. R is the gas constant, T is the temperature in Kelvin, and Q is the reaction quotient, which is calculated just like the equilibrium constant K, except that the concentrations are not at equilibrium. For example, for the reaction A plus 2B goes to 3C, the reaction quotient looks like this. Therefore, it takes into account the concentrations of all of the species in the reaction. This is how delta G is calculated for most chemical processes. However, biochemical processes are slightly different, and so we use a slightly different nomenclature. In biochemistry, we use delta G prime and delta G naught prime. The prime indicates that we're using standard biochemical conditions rather than standard chemical conditions. This results in slightly different values of delta G and delta G naught. Specifically, the differences that we're including in this prime value are that the concentration of water is 55 molar, which it is in all biological systems, and that the hydronium ion concentration is 10 to the minus 7th molar. In other words, the pH is 7. In this course, we will always be talking about the biochemical standard conditions. Sometimes, however, we'll use delta G and delta G naught without the prime, but know that they mean the same thing, specifically under biochemical standard conditions, which will be what we use for this course. The calculation of delta G prime is useful because it tells us the driving force of a biochemical process. We know that the reaction will proceed until delta G prime becomes 0. In other words, the driving force of the reaction becomes 0. When delta G prime becomes 0, the reaction above changes. We can rearrange this equation to give this. When the reaction reaches a delta G prime of 0, we say that the reaction is at equilibrium. At equilibrium, Q is given a special designation, K. K is calculated in the same way as Q, but it is only when the reaction is equilibrium that we call this value K. Q and K are useful because a comparison of these values can tell us if a reaction is at equilibrium or not. If Q and K are equal, the reaction is at equilibrium. If Q is less than K, we can expect that the reaction will be spontaneous in the forward direction. Conversely, if Q is greater than K, we can expect that the reaction will be spontaneous in the reverse direction. Thermodynamics is useful for predicting which direction a reaction will proceed, but it tells us nothing about how quickly the reaction will occur. This is kinetics. Kinetics is the study of how quickly a reaction will occur, in other words, the rate of the reaction. Biological systems can also control the kinetics of reactions. Specifically, they use enzymes to speed up the rate of reactions. They do this by acting as a catalyst. Specifically, they lower the activation energy of the reaction. The activation energy is the energy of the transition state of the reaction. The transition state is the highest point of energy throughout the reaction process. By lowering this energy, enzymes can speed up the rate. We can visualize the thermodynamics and kinetics of a process using something called a reaction coordinate diagram. A reaction coordinate diagram graphs the energy versus the point in the process or reaction, otherwise known as the reaction coordinate. In the process of A being converted to B, this is the reaction coordinate diagram. On the y-axis, we have free energy. And on the x-axis, we have the point at which we are in the reaction, or the reaction coordinate. We go from left, reactants, to right, products. This reaction coordinate diagram shows two processes. In red, we have the catalyzed process, and in blue, we have the uncatalyzed process. In the uncatalyzed process, the energy of the transition state, the highest point in the reaction, is higher than in the catalyzed process. This is due to the catalyst, or the enzyme, which lowers the activation energy, making the rate of the reaction faster. Notice that catalysis, in this case going from blue to red, changes the rate of the reaction, or the energy of the transition state, but it does not change the overall thermodynamics of the process, which is the delta G value, which is the same for both the blue and the red. The energy of the reactants and the energy of the products do not change. Therefore, the difference in energy between them does not change, even in the case of catalysis. Adding the enzyme only changes the activation energy and the rate of the reaction. This process is exergonic because the products are lower in energy than the reactants.